Zebrafish is a tropical freshwater fish originally from South Asia that you might already know because it's a popular aquarium fish. Christiane Nusland-Vollard was really the one to bring zebrafish to the stage as a model organism when she did large genetic screens in the 1960s in Tübingen, Germany. Historically, this is a much shorter time compared to using mice or fruit flies in research. But the zebrafish with its external fertilization has really allowed us to learn lots of information when it comes to the early stages of development, namely fertilization. Additionally, because of its small size, it's been very useful in chemical screens. Today, Anna and Nancy are going to give us a tour of the zebrafish facility at Forskningsparken here in Oslo. Hi Anna, this is Ricci.
Welcome girls. Hi Viola, hi Ricci, nice to meet you.
So you're going to tell us about zebrafish today?
Yes, of course. I'm going to answer all your questions about the fish. Here in the rack number 1, we are hosting the fish. We have them to produce embryos. The embryos that are being used by the different researchers in different projects. Here is another rack hosting mostly the immune lines.
And so do you start, I see there's different ages, do you start working on them as juveniles?
Yes, they are starting first in the larva bench. We are keeping them until 21 days, jumping after to the baby rack when we are controlling the stream because they are not developed enough to swim against the current. And after, from those small tanks are starting to reach maturity, so they are being separated in females and males in different tanks. So they are getting sexually mature to start producing embryos.
So what's the most important parameter to control when you take care of zebrafish?
The water quality. We have been investing a lot of time and research to give the best conditions for the fish, not only about the temperature or the pH, but also the hardness and also the subproduct of the fish metabolism to keep this in balance, having our systems supported by the biofilter. So if you're asking who is living in your system, it's not only zebrafish, it's the complete symbiosis with the good bacteria.
Very good. So it's hot in here. What's the temperature?
Let's see.
So
now it's
about
28
yeah.
degrees.
The tropics. So can you show us how you would set up a cross to get some embryos?
Yes, of course. Here is one of the special tanks. The special tanks have this kind of geography, being different for them to imitate nature and allow them to spawn in the shallow water. What is happening in here? We have been separating both, the males and the females, and divide by a barrier. We are setting up this breeding tank the day before. And during the night cycle, they start to exchange pheromones. The female release a pheromone that is telling to the male, follow me, is starting the chasing behavior, while the males swimming in groups around them release the ovulation hormone. So once that they reach the shallow waters of the river, they are ready to release the sperm and the eggs to produce fertilized embryos.
So can you just point out which ones are the males and which ones are the females?
Yes, it's easy to see that the female have a little bit more rounded figure. It doesn't mean that she is fat, but she needs a space for the eggs.
Exactly.
While the males are a little bit more pinkish and more slender in figure. Yeah. You can see the difference, Ricci?
Yes, I do.
The skinny males. So then once you've obtained embryos, then I hear you have an injection apparatus.
Yes, please follow me and I will show you the embryos and the setup for the microinjection. Girls, meet Nancy.
Nice to meet you, and you're welcome to my workstation today.
Great.
So join me under the microscope. So under the microscope, I have embryos that were born today. They were born about 3 hours ago. And for zebrafish larvae, after like 30 minutes, they already start going to cell division. So at 30 minutes post-fertilization, usually you have the one cell stage. And then about every 15 minutes, you see duplication of the cell. So from one cell stage, you have 2 cell stage, 4 cell stage, and etc. Already by 24 hours, then you already see the full body layout of the fish, and that is something that is so fast and you don't see in other animals. For instance, at this stage you don't see that rapid movement. You see the rapid movement at 3 days old. That is when it is thought or is believed that the motor neurons are well developed by then. But here you still see them moving a bit, but not so much at this stage.
So one of the methods then that is used a lot, especially with zebrafish, is then microinjection, and I know that you use this as well. So can you show us your setup?
Yes. So in this lab, we have the micromanipulator set up, but then for some reason we feel that we are faster when we do it by hand.
Wow.
So I would demonstrate to you how we do the injections. So we need, first of all, like a needle. So this is like a capillary. And then, so for instance, this morning I did morpholino injections. So for morpholinos, those are like oligonucleotides that are used to knock down specific genes. So this method does not lead to permanent changes to the genome. It's only temporary, and usually between 5 to 7 days, then the morpholino effect would have faded out. This is the setup. So this is like the femtojet setup. So you have the pressure, you have the time it takes, and then we put this compound we want to inject in the needle.
So you inject the yolk then, you don't inject the
yeah,
cell?
For morpholinos, we inject into the yolk.
What do you think, or what are the strongest advantages with working with zebrafish?
First, my favorite admiration for working with zebrafish, the fact that it's easy to handle. I have worked with mice and rats before, and I can tell you it's a lot difficult, even picking them. But with zebrafish, it's it's a lot easier, and also the space that they use is very small. So in terms of running cost, it's cheaper. And secondly, they are small in size, and they are able to fit into like 96 well plates. So at like 5 days old, they are about 4 millimeters long. So they are able to fit into these tiny well plates, and then you are able to put them under like a microscope or a camera, and then you can observe them in detail. Another thing is that they are optically transparent up until like 7 days old. And because of that, we're able to do a lot of in vitro. We don't have to euthanize them before we go to slice their brains to find specific areas. But under the microscope, until 7 days old, we are able to just work with them whilst they are alive. Also, they are genetically tractable. That means that most of the tools that we need to edit the genome, it's in zebrafish are available. So if you find a gene that you are interested in working with, you can easily kind of edit the gene, for instance, and then you can work with them. And another advantage, considering that we are a chemical neurobiology lab, is that they absorb substances like drugs through the water. So you just dissolve the compound in the water, and then the fish absorbs the active compound, and you can easily read out the effects. And one of the last points I would like to mention is that fertilization occurs ex utero, and development also occurs ex utero. So for instance, usually when you're working with mice or higher vertebrates, when you want to modify the genes, you need to perform like a surgery on the mothers, take out the eggs, edit them, put it back, and this is very stressful. And if you're not very experienced, you may end up killing the mothers. So these are some of the advantages.
So what does Camila Esguerra's lab focus on, and what does your project focus on?
So the focus of the Esguerra lab is to understand the mechanisms of brain function in the healthy state and in the disease state. And specifically in this lab, we are interested in understanding the mechanisms of seizure generations and also pharmacoresistant epilepsy, for instance. But recently we have also moved a little bit away from just epilepsy into looking at other neuropsychiatric disorders such as schizophrenia and autism.
One of the assays the Esguerra lab uses to study neurological disorders is the analysis of swimming behaviour, and Nancy is going to show us a machine called the Zebrabox that allows these types of experiments.
Wow, it's hot.
Yes, you're welcome to the Zebrafish behavior room. And as you exclaimed, it's really hot in here because the temperature is 27 degrees constant. So I'll just briefly take you through some of the behavioral experiments that we do. One is the tracking, which is just a simple locomotor assay where we track whether the fish have a normal movement or behavior versus maybe mutants or drug-treated fish. And also I would show you an example of a behavioral test called shoaling. Just like humans, zebrafish also want to be together. They don't want to live in isolation. So when you put them together, then you're able to monitor how close they stay together and how far they are. And it's well shown that in zebrafish, when they are given like alcohol, like humans and stuff, they tend to behave in a certain way and vice versa. So since all our parameters are set, so you see the tracking of the fish. This fish is not moving, so it's not being picked up. And this is really moving. So in this test, I have 5 larvae in each arena. Okay, so it's tracking them now. And then we're going to measure the time that they spent together. And then what this does is that it picks one fish as the reference, and it makes kind of like a quadrant, how far each fish is from that fish.
Yeah. I have a huge positive bias to the zebrafish because it is a model organism I have worked with myself, and it has remained my favourite model organism. I think the fact that you can observe development live under the microscope, combined with the fact that it is a vertebrate, is absolutely fascinating. Today we have learned about how to cross fish, how to manipulate embryos, and to do behavioural analysis. This tour has been excellent.